G'day everyone. Just while folks are joining, I hope you're enjoying this hypnotizing video of a fungus under the microscope. This is the glues and gums that make the crumbs of soil aggregates. And if there's one image that we could have you take away from this webinar, it's this image of fungus growing through the soil, forming soil aggregates, because inside soil aggregates, there's a slower turnover of carbon and nitrogen in soil, while of course aggregates are the inverse of porosity. And so with improved aggregation, you'd have improved water infiltration and retention in soil. So this is a webinar on CarbonBuilder from Loam Bio. The reason we're here is that over the last century, we've seen over 50% of soil carbon lost from agricultural soils. And at the same time, there is 9 gigatons of carbon dioxide emitted in the atmosphere every year. And so there's clearly a mismatch here between soil carbon, which will drive soil health and productivity and nutrient availability. There's a gap, and there's too much carbon in the sky at the same time. So my name is Robbie Oppenheimer. I'm the Chief Product Officer with Loam Bio, and this webinar will be about CarbonBuilder, the fungus that we've developed to increase and stabilize soil carbon. Many of the people on the call, farmers or agronomists, are interested in soil health because that's a driver of productivity, nutrient availability, water retention, and ultimately yield. And you'll know that we have ways of increasing soil nitrogen through chemical fertilizer or nitrogen fixing microbes, ways of increasing phosphorus through chemical fertilizer or phosphorus solubilizing bacteria. But what tools do we have in the toolbox for increasing soil carbon? And that's why we've developed a microbial product for broadacre that addresses this problem. Maybe one of the thoughts or objections you have in your mind coming into this is that what if this fungus doesn't work on my farm? So we've run over 60 field trials across Australia, and in fact we've seen 3 to 5 tons of carbon dioxide per hectare increases, as well as some small increases in yield in locations spanning Western Australia, Victoria, New South Wales, and Queensland. So we believe it will work in your area because we've got the data. And in this webinar, we'll spend 45 minutes walking through what the product is, what results we've seen, how we developed it, how it works, and how you can use it. And there should be some time for questions at the end. So as I've said, the main problem here is that agricultural soils have lost carbon, resulting in lower productivity and the requirement for increasing inputs. And as we look forward over the next 20, 30 years, we know there's going to be increased variability in climate that we're already experiencing. And soils and having healthy soils plays a key role in that, by buffering against these external shocks. This also represents a massive opportunity. If we can sequester that carbon, we can increase the efficiency of our farming systems, requiring fewer inputs. And this also creates opportunities for business resilience. That's resilience to those climate variability, but also if there's the potential to capture the value of that sequestered carbon through insets or offsets in carbon markets, creating a new revenue stream for farming businesses. And so if you've thought about this or talked to anyone in the space before, you will have seen or experienced skepticism around whether it's even possible to build soil carbon. And people are right to question that, because if you look at the data, if you look at an example, here's a 16-year trial of different farming systems in Australia. What people have observed is an increase in carbon over the first 13 years in these 4 different farming systems: continuous cropping, conventional tillage, perennial pasture, and reduced tillage, and subsequently the loss of that carbon. All of these different farming systems ended up with lower TOC, except for perennial pastures, which ended up back where it was 16 years later. And so what's really interesting and insightful, if you dig through a paper like this, if you go through the detail, they also indicate why this happens. Some of the specific parts which are interesting are noting that the carbon to nitrogen ratio in that soil increased from about 14 in 1999 to about 19 or around 20 in 2012 before this subsequent collapse. And why that's interesting is that the natural carbon nitrogen ratio, or the equilibrium that it seems that soils push to globally, is around 11 to 13. So this is a sort of warning sign that the soil system is getting out of balance, and that excess of carbon will be lost as the microbes churn through and respire that carbon. What's also interesting in this long-term study is the splitting of a soil sample into the different component fractions. They saw an increase in particulate organic carbon over that period from 14% to 40%. So it started off about 14% and it ended about 14%. That's this plot here. You almost can't tell the difference in this plot on the right, even though it's actually changed from the total organic carbon, TOC, to particulate organic carbon. So particulate organic carbon is the large and light material in a soil sample. It's plant biomass, roots, leaf litter, insects, debris that are readily available to be digested and turned over by microbes in the soil. So taking these 2 points means that the actual challenge is not building soil carbon. Like, it's possible to increase. The challenge is stabilizing the soil carbon. The challenge is focusing on the pools of carbon, like carbon in soil aggregates and carbon on mineral surfaces, where it will be stable to changes over time. So this is a view of the carbon cycle. Obviously, plants photosynthesize, pull carbon down into the soil. This can happen within hours. Carbon is then respired back into the atmosphere by plants and microbes in soil. A lot of work and a lot of interventions that people make are simply about putting more carbon into the system. And the insight that drives Loam's innovation actually came from Professor Peter McGee at Sydney University, which was that simply adding more biomass or compost or manure can perversely stimulate the microbial community in soil and actually end up in more carbon being lost. The intervention that makes the most difference is focusing on how you stabilize carbon in soil, and microbes play a key role in that. In particular, Professor Peter McGee focused on a family of organisms called dark septate endophytes that grow associated with roots and access plant carbon before it gets respired back to the atmosphere. And as the fungal hyphae grow through soil, they can pull soil particles together into soil aggregates. If carbon is inside that soil aggregate, it's then less exposed to oxygen and other microbes that will decompose it and respire it back to the atmosphere. So it's really about these two chemical processes that are responsible for the decomposition of carbon. This is getting into the sort of high school chemistry. It's limiting oxidation and hydrolysis of soil carbon. And this family of organisms, dark septate endophytes, do those two things: reducing oxidation by putting carbon in soil aggregates, and reducing hydrolysis through some of the compounds that they produce, the melanins and polyaromatics that are less susceptible to hydrolysis, and that's how they can slow the turnover of carbon in soil. So, jumping back, what does Loam do in the face of this challenge? We have a CarbonBuilder product. It's a world-first microbial inoculant to stabilize soil carbon, and we also partner with farming businesses to run carbon projects, and that's one way of capturing the value of that sequestered carbon. We haven't developed this product in isolation. I mentioned that the original product concept came from Sydney University. There's also been a range of academic collaborators that we've worked in Australia and internationally to develop this product concept and to show that it worked in a range of systems. There's also been a variety of investors in Loam that have seen this opportunity and seen how unique this technology is as a solution. We've taken that support and walked through this process to develop the microbial inoculant. First was the isolation of a native microbial library. So there are over 2,000 organisms in our Australian library that have come from soils all around Australia. These are natural organisms. They're not genetically modified. They're not modified in any way from their initial condition. We've then identified the organism and selected those that we believe have the highest potential for carbon sequestration. There's been a range of laboratory studies testing compatibility with the fungicides, insecticides, herbicides that these organisms need to be resilient to if they're going to survive in a real system, and also doing our first screens for the performance traits that are associated with building soil carbon. So can it aggregate soil in a petri dish, as an example? The first real test of whether it can increase soil carbon and improve plant performance is then in a greenhouse setting. And so that's work that we've done internally, but also through our academic partners, before then proceeding to a range of small plot trials and strip trials at locations around Australia. Once something is validated in the field, there's also the large-scale production and manufacturing and quality control to ensure that every batch of CarbonBuilder is the same. So that's the process that we've walked through. Coming out of that process, we have 3 products in Australia: CarbonBuilder wheat, barley, and canola. In the US, CarbonBuilder soy and corn. And we're currently working on CarbonBuilder pulses for Australia. So this is the overall mechanism. Carbon dioxide is drawn down from the atmosphere through photosynthesis, circulates through the plant into roots in a matter of hours, feeding the microbial community that live around the plant root. Root colonizing fungi use that carbon to explore the soil environment. And as they reach out, they bond together soil particles into soil aggregates. So these fungi can colonize both the outside and the inside of soil aggregates, and in that way stabilize carbon inside aggregates and on mineral surfaces. So the clays, the iron oxides, the aluminium oxides in soil. And that's where carbon is actually most stably bound. So one point that I'm trying to communicate in this webinar is that not all soil carbon is created equally. We spoke earlier in that long-term study about particulate organic carbon. That's the roots and leaf litter and material. This is the least stable. It can turn over in weeks to years, depending on the temperature, moisture, available nutrients. So if you are building particulate organic carbon over time, there may be useful agronomic benefits. There may be useful reasons why it's good to have higher biomass, but you can't expect that to be around long term. For example, if someone had a carbon project and particulate organic carbon was increasing over time, you should expect that to be lost. This would be a risky way to increase soil carbon. The other pools of soil carbon and mineral associated organic carbon is probably the exact opposite of particulate organic carbon. This is the smallest and densest material. So this is the clays, the aluminium oxides. When carbon gets bound to that surface, it's then unavailable for microbial respiration. And so that can take centuries to turn over. This is based on carbon isotopic studies where people have taken carbon dioxide labeled with an extra neutron, so C13, and be able to look at the turnover time of that carbon in soil. This intermediate pool, called occluded particulate organic carbon or aggregate carbon, can take decades to centuries because inside those soil aggregates, it's protected. Carbon is protected from oxygen, and other microbes. This is one that we focus on because there's also other co-benefits of soil aggregation in the nutrient use efficiency in soil, water use efficiency in soil. I'll speak to some of those now. This is an example of sterilized sand. This is a purely proof of concept laboratory experiment. On the right, it's sterilized sand where we've added water, and you can see that water flowing through the sample. On the right, this has been inoculated with a fungal isolate from our library at a very high dose rate. The fungus has grown through the soil and changed the physical structure. When the same amount of water is added, that water is retained in the soil sample. So this indicates that the mode of action that fungi can enact on soil changes the physical structure and the ability to absorb water and to hold nutrients and carbon. This is an example of that in practice at a strip trial. So this is a 2-year strip trial at the Dimboola trial farm. It has 5 treated strips, 5 untreated strips side by side. In 2023, it had CarbonBuilder barley applied. In 2024, it had CarbonBuilder canola. After harvest in 2024, OptiSoil took soil samples from that trial and analyzed them. This is actually, just to give you a picture of it, so that everyone knows what we're talking about, this is actually this trial. It's the same one as this picture. We've got treated and untreated strips here for canola. And what they did is take those intact soil samples back to the lab and then measure the rate of water infiltration through those soil samples. Interestingly, after 2 years of application, they saw a statistically significant increase in the water infiltration rate at both the 0 to 10 and 10 to 20 centimeters. Another way of looking at this is in same strip trial design, but looking at the ability of soil to hold on to water, not just for it to infiltrate. So same trial design, but in Victoria, 50 soil samples sent to the New South Wales DPI to assess water holding capacity. So in the soil samples, we measured the permanent wilting point, which is the sort of lower limit for available water, where basically a clay would hold onto that water so tightly that it's not available to a plant, and measured the upper limit, the field capacity. So that's the point where a soil sample is completely saturated and you can't add any more to it. The difference between those two is the plant available water, which will vary at different points in the season depending on the amount of rain. In that trial, there was a change in the plant available water in CarbonBuilder treated strips. There was no change in plant available water in the control strips. That 1% increase in plant available water corresponds to effectively an extra 10 mils of rainfall if that occurred all the way through to 1 meter depth. Some of the work that's been done from our academic collaborators has also looked at nitrogen. So the University of Western Sydney ran a study where they played with 12 fungal isolates from Loam's library and looked at the effects on carbon, nitrogen, and plant growth. In this case, for 2359, which is the active ingredient in our CarbonBuilder wheat product, they saw at the end of the trial 8.4% higher total nitrogen in the pots that had been treated with 2359. And interestingly, they saw 29.5% higher nitrogen in the soil aggregates. And what's actually really interesting about this specific trial is that it started off at 0.39% nitrogen. So just so everyone's on the same page, the initial conditions for this growth chamber trial were actually up here. It lost nitrogen in the uninoculated control and held on to nitrogen in the 2359 treated strips, treated pots. How did it hold on to more nitrogen? Because there's more nitrogen in aggregates, where it is less easily lost through respiration and leaching. That's an important bit to remember is looking at this, that obviously, wheat can't fix nitrogen. A fungus doesn't fix nitrogen. Where does this apparent nitrogen come from? It's not through an increase in nitrogen, it's through stabilizing nitrogen against loss. A similar effect was observed from the University of Queensland. This paper has come out earlier this year in a soybean field trial, the same isolate. They saw 20% higher total nitrogen at harvest, with more nitrogen in soil aggregates and also more nitrogen on mineral surfaces as well in this trial. So to try and pull some of this together to give you a clear story, CarbonBuilder's mode of action is to aggregate soil. And that has a number of indirect benefits on carbon cycling, nitrogen cycling, and water cycling. It affects carbon through the formation of stable microaggregates that slow the turnover of carbon in soil and reduce the loss of carbon in these labile fractions of particulate organic carbon. The net result is being able to build soil carbon over time. The aggregation of soil can also affect nutrient use efficiency. One of those direct mechanisms is the root-associated fungi trade nutrients for carbon with the plant. So if the plant photosynthesizes, it gives that carbon to these root-associated microbes in exchange for nutrients that they're pulling from the soil. We've also just seen on that previous slide that it's possible to reduce or to slow the turnover of nitrogen in soil and reduce the loss over time. The net outcome that we're excited about is the possibility of lowering emissions intensity of our farming systems. That final point about aggregation is that it affects porosity, root access to water, and the ability to hold water in soil. This gives us some climate resilience. As we know, we're going to experience increasing fluctuations in temperature and moisture. I'm going to shift gears now to go from some of the work from academic partners to some of our own field trials and specifically what that looks like for carbon. I mentioned early on that there were over 60 small plot trials in locations across Australia. This is a summary. If we take those small plot trials and take the average increase in carbon that we see, the average increase in yield that we see over 3 years for 31 field small plot trials in a range of locations and soil types. Similarly for wheat and similarly for canola. On average, what this corresponds to is about 3 to 5 tons of carbon dioxide equivalents per hectare and a small increase in yield. This is jumping back to that strip trial that we saw before, where there was the increase in water infiltration. In this trial, so this is the second year of application, it had CarbonBuilder barley in 2023, CarbonBuilder canola in 2024, run by Pure Grain and their team up at Dimpler Trial Farms. In this case, as you can see from the drone image, an increase in biomass in every treated strip compared to the untreated strips. What we see for carbon results for that trial over time in 2023, an increase compared to control, both the control and the CarbonBuilder increased. But we can see in the CarbonBuilder strips an extra 5 tons of carbon dioxide equivalents per hectare. And what's interesting in this specific trial is that this was a dry year. So there's reduced moisture, so there's capacity for that carbon to sit there waiting for moisture before it's respired back to the atmosphere. That's actually what we saw in 2024, higher rainfall year, and the uninoculated control lost carbon in that period. The CarbonBuilder treated strips stayed the same. So they retained, compared to control, 4.4 tons of carbon dioxide equivalents per hectare. These are the yields in that trial. In that first year, 2023, an average 9.8% higher barley yield with statistical significance. This was in that dry year, and we saw a clear treatment effect. This is also kind of what you'd expect in a time when a plant is struggling. They tend to send out signals to recruit microbes and feed carbon into the microbial community. In 2024, wetter year. In this specific case, it was nitrogen limited. So even though there was the increase in biomass, it didn't translate to higher yield. Slightly lower yield in the CarbonBuilder treated strips without statistical significance. At the same trial farm, there was another trial running in 2024 with barley. In this case, no significant effect on yield, but an increase of 3.9 tons of carbon dioxide equivalents per hectare. So in this location, the uninoculated control was decreasing in carbon over this period. The CarbonBuilder treated strips increased. Third trial at the same location with wheat in 2024. In this case, there was actually no harvest results. There was an error during harvest. The tons of carbon dioxide equivalents, so CarbonBuilder treated strips maintained carbon or slightly increased carbon over that period, while the uninoculated control lost carbon at that site. So again, this is pointing to the mode of action that CarbonBuilder stabilizes against loss of carbon by putting that carbon into these more stable fractions, aggregate mineral associated pools. This is another example of a 2-year strip trial, the same design. So this is a 40-hectare paddock in Forbes, 5 treated strips, 5 untreated strips. In 2023, it had CarbonBuilder canola. In 2024, the treated strips had CarbonBuilder wheat. So in this case, what we can see is an increase in both control and CarbonBuilder for carbon over that period, but a greater increase in the CarbonBuilder treated strips. What I'm really interested to see with this trial is when it hits the right season, when it hits the right point. The prediction would be that the uninoculated control will lose carbon and it will maintain that carbon in the treated strips by stabilizing carbon in the aggregate mineral associated fractions. At this location, 2023, there was an 11% higher canola yield. In 2024, almost 4% higher wheat yield, not statistically significant in either of those. This is a trial run by Outlook Ag, and they did a piece of analysis on this, which has been really insightful and something that I now want to do on all of our strip trials. So same strip trial design, treated, untreated strips, more than 30-hectare paddock in Balata, but they broke it down into the different soil types within that paddock using this red, green, blue index from satellite mapping. So basically what they can see across this area is a mix of heavier soils, higher clay content, to lighter soils. When they split out the results here, though there's an average 14.5% yield increase across the paddock, there's a small increase in yield in the heavier areas and a much larger increase in yield in the lighter areas. And again, this ties to that point about when plants are struggling to be able to access the nutrients or water that they need, they tend to interact a lot more with the microbial community to access those nutrients in soil. So in a way, this creates a kind of resilience or buffer against loss of yield in certain areas of the paddock. One of the questions you might be asking is, that academic work on these different soil carbon fractions, do we actually see that at full farming scale in a normal business as usual situation? And that's what we can see in this strip trial. So this one is run in Elmore, Victoria. This is the same trial that I shared earlier that had the increase in plant available water of 1%. So at that location, 40-hectare paddock, there were 100 soil samples taken across the paddock, 50 in the treated strips, 50 in the untreated strips. The total tons of carbon dioxide equivalents per hectare, total carbon stocks, the same at sowing. At harvest, there's an increase in carbon for both the unallocated control, but a larger increase for CarbonBuilder, which corresponds to an extra 7.4 tons of carbon dioxide equivalents. We took those soil samples and split them into the different component fractions. The mineral associated fraction, MAOC, which is most stable in green, the aggregate carbon, and the particulate organic carbon. So here at sowing, you can see that there's no difference between the treated and untreated strips, and that makes sense because that's just before the application of CarbonBuilder barley at sowing. At harvest, there's a large increase, which corresponds to the total carbon that we see on the left. And a lot of that change has come from the particulate organic carbon. And that makes sense because we've just grown a plant through that period. There's all of the plant biomass and the roots and the root exudates. That particulate organic carbon has doubled from sowing to harvest. However, it's doubled in both the uninoculated control and in the treated strips. And the anticipation, just like we saw in that long-term study over 16 years, that increase in particulate organic carbon is going to evaporate. That's the quick turning over pool of particulate organic carbon, which is going to be lost back to the atmosphere. What's really exciting in this trial is seeing a 7.2% increase in aggregate carbon and a statistically significant 10% increase in the mineral associated carbon, that most stable fraction. Previously, I spoke about the University of Queensland work that was published earlier this year. What's interesting in this trial is they saw the same kind of data where there's an increase in total carbon with our 2359 strain, that's the active ingredient in CarbonBuilder wheat, 13.2% higher levels of total carbon. But what's driving that ultimately is the increase in aggregate carbon, the increase in mineral associated carbon. And what's exciting about in this trial is also they saw a large increase in grain yield, more than we typically see in our own trials. At the same time from that location, and one thing that we've learned from these academic collaborators is also to focus on nitrogen, because they saw an increase in total nitrogen, increase in aggregate nitrogen, 20% increase in nitrogen in the mineral associated stocks. The important point when looking at this is to say, how does this affect the carbon to nitrogen ratio? We don't want to make that same error that we saw in the long-term study at Condobolin, where there was an increase in carbon nitrogen ratio and then it was all lost. So actually what was observed here is that the plots that had the higher dose rates of 2359 actually lowered the carbon nitrogen ratio, got it closer to the around C to N ratio of 12, which is the natural stoichiometry that soils are pushing to. And what this means is that while carbon and nitrogen both increased, the nitrogen increased more than carbon in this case. So we've just gone through a whole set of data. So to jump out from that for a moment, want to talk about how CarbonBuilder is actually applied. So this is an example at full scale, because applying a seed treatment at sowing would be challenging and would has the potential to add a step. So to improve the convenience and make it as easy as possible to use, we've developed Furomate. So this is a unit that straps on the side of an air seeder and has a little auger that can dose out the dry powder of CarbonBuilder into an air seeder. So this is the Furomate unit that was strapped on the side of the air seeder. It's a direct air injection unit and basically forms a practical solution for applying dry powders, which in this case is the CarbonBuilder containing the fungal active ingredient, the fungal spores. That dosing meter connects to a unit in the cab so that a farmer can dose in exactly how many grams per hectare they need of the CarbonBuilder powder, while there's a blending unit that can turn over that powder in situ to ensure that it's evenly dosed out. This is an example of what that actually looks like as the powder is filtering through that auger. So if you'd like to learn more about the Faramate, learn more about CarbonBuilder, learn more about carbon projects through Second Crop, this is the team that you can speak to. You can call us on this number, email us here, or speak to any of our team in your area.